بسم الله الرحمن الرحيم الحمد لله رب العالمين والصلاة والسلام على أشرف الأنبياء والمرسلين وعلى آله وصحبه أجمعين رب اشرح لي صدري ويسر لي أمري واحلل عقدة من لساني يفقهوا قولي. Assalamu alaikum warahmatullahi wabarakatuh and very good day to everyone. So let's continue the second part of chapter 5, which is immune response. So last week I already covered phase of humoral immunity, the activations of humoral immunity systems until activations of the lymphocytes. So today we're going to continue on clonal expansions and differentiations, stage number 3, and the last one is effector functions. And later on we covered the phase of activations of antibody, and as well as activations of cell-mediated immune response. So right. So the third stage in humoral immunity activation is clonal expansion and differentiation, which is, as I told you last week, where the B cells will be activated into plasma or memory cells after the first infections. Okay, so in this clonal expansion and differentiation, the transcription factor is activated, produce a protein that involved in the B proliferations, right, which is clonal expansions, and differentiations. B cell differentiate into effector cell, which is plasma cell that secrete antibody, and the memory cells. So it means that for clonal expansion and differentiation, the naive B cells, that have been recognized the antigens with the help of T helper cells. All right, so T helper cells actually play a big role to mediate the both immune system response, which is cell-mediated as well as humoral response. So T helper cell also will send signals toward naive B cells in order for the B cell to be differentiates and proliferates into plasma cell that secretes antibody. This is the antibody factory, and as well as the memory cells. If there is a second infection, so memory cell will be activated. Okay, so once activated, the B cell may generate up to 4,000 antibody secreting cell, which can produce up to 10 to the power of 12 antibody molecule per day. So it means that B cells, plasma B cell, is a very powerful, I can say, a very active factory that keep producing the antibody towards our systems. Okay, and the secreted antibody have the same specificity as the naive B cell membrane receptor that recognize antigen to initiate the response. Means that the antibody that been produced by plasma B cells is the same as it has the same specificity as a naive B cell that been recognized the antigen before. Okay, so that the antibody is very specific toward the specific antigens. Okay, and repeated expose to a protein antigen results in the productions of antibody with increasing affinity for the antigen affinity maturation. So let me change this one first. Okay. Right. Then the last factor is factor number 4, which is effector functions of antibodies. Okay, so when B cells is being differentiated and proliferates into plasma B cells and memory B cells, then the antibody being produced by plasma B cell now can be activated. Right? In other words, mean that it can recognize antigens or bacteria independently. Right? So antibody do not kill or eliminate antigen. So this is a very, I can say, a cliche definitions. So that because most of students or sometimes publics saying that antibody can kill bacteria, antibody can kill viruses. So it's not meaning like that. So antibody only helps, all right, the bacteria or the viruses or the antigen to be killed by killer cells, all right, which is undergo phagocytosis, for example, macrophage, dendritic cells, or natural killer cells. Okay, so their roles are to bind to antigen and mediate effector functions. Okay, and the binding of antibody to antigen can result in several mechanisms. So basically, there is 5 mechanisms that can be mediated by the antibody, which is the first one is agglutinations, the second one is opsonizations, the third one is neutralizations, the fourth one is activations of complements, and the last one is antibody-dependent cell-mediated cytotoxicity, or known as ADCC. Right. So we're going to I'm going to explain each mechanisms. So the first one is agglutinations of microorganisms. Right. So the antibody in agglutinations of microorganism is basically the antibody that being produced by the plasma membrane, so by the plasma B cell. We target the specific, for example, here bacteria, right? So the antibody will capture the bacteria, and the bacteria being captured become agglutinates, right? So it become bigger, so that it can be easy for the macrophage to recognize and kill the bacteria by phagocytosis. Okay. And the second one is complement activations. Okay? Complement activations means that formations of immune complex can trigger complement cascade reaction, which can lead to formations of pore and cell lysis. So complement activation basically can be classified or can be grouped into 3 pathways. Okay, so the first one and very distinctive pathway is classical pathway. The second one is MB-lectin pathway, and the other one is alternative pathway. So yeah, the mechanism is so complex because it's involved like more than 20 proteins to ensure that the bacteria, the pore of the bacteria become not visible, become easy to get invade. The protein can invade into the bacteria membrane cells and force the bacteria to undergo lysis, exactly like this. And the third one is opsonizations and promotions of phagocytosis. Okay, antibodies coat microbes and promote their injections by phagocytosis. So the opsonization is almost the same as agglutinations. Okay, but then in opsonizations, one bacteria can be recognized by several of antibody, right? So by doing this, it can enhance phagocytosis, right? So that bacteria phagocytosis to kenal, it can recognize the bacteria that already been covered by antibody. Okay, and the fourth is neutralizations of microbes and microbial toxins. Okay, so in these neutralizations, antibody bind to and block, or in other words, neutralize the infectivity of microbes and interactions of microbial toxins with host cells. Okay, so in these neutralizations, bacteria can either recognize a single particles or single virus or single bacteria, or it can also recognize the toxin that been produced by the bacteria. Right? For example, you have this bacteria A that invade into your systems. So the bacteria itself will release such a toxins that can, alter the host cells, compositions, for example. So those antibody can bind to the toxin as well. Okay, so by binding into the surface of a bacteria, virus, of the toxins, it will help to stop the bacteria or the antigens to invade into the host systems, right? In other words, that this bacteria is neutralized, it's not functioning because it cannot invade into the host cells. Okay, and the last one is antibody-dependent cell-mediated cytotoxins, or known as ADDC. Okay, so the linking of antibody bounds with the target cell with the FC receptor can induce or direct the cell cytotoxins activity of the effector cell against the target cell, lysis of the target cells. So it means that mainly the antibody that involved in ADCC is IgG, right? So IgG will binds to the, for example, antigens, the surface of antigen, right? So that, for example, this is bacteria or a parasite. Okay, for example, a parasite worm. On the surface of the bacteria of the parasite, they have epitopes of antigen here. Okay, so the antibody IgG can recognize it. Okay, and for example, NK cell, natural killer cell, will recognize the IgG that bind to the antigen surface. Okay, so the NK cells, after it recognize the antibody bind to the antigen, it will release cytokines, right? Signals, specifically known as interleukin 2. Okay, so this interleukin 2 will react against the bacteria, right, to undergo cell lysis. Okay, and special types of ADCC mediated by eosinophils play a role in defense against Helminth's infection. So ADDC works for normal antigens, and it also works well for parasites. For example, Helminth's infection, worms. Okay, and the last one is ada juga number 6, immobilizations of bacteria and protozoa. Okay, so mostly bacteria and protozoa, they have flagella or cilia, right, to help them to move around. And the antibodies can actually bind to the cilia or flagella, thus the structure will be less active, right? So easier for phagocytes to engulf. Right, so this is test yourself. You can do it by yourself. Right, not to be continued. Okay, let's continue to the types of humoral response. Okay, so antibody response to the first exposure to an antigen is called primary immune responses. Okay, so primary means that first exposure lah, and the subsequent exposure are called secondary immune responses. Okay, so when you have like first attack, the immune system will react. They call it primary immune response. But when there is a subsequent infections, the same, for example, antigens, okay, so we call it a secondary immune response because the second time the immune response will be react. Right. During the primary immune response, immediately after the first exposure, the antigen there is a duration called the lag period. Okay, so just ignore the second part one. So let's focus on the first part of the diagram. Right. So when there is a first exposure, the antigen introduced into our body, there is a lag period here. Okay, so the lag periods occur because the lag period occur because when our adaptive immune system is being mobilized, all right, and appropriate lymphocytes are being cloned to that particular sites. Okay, so which means during the infection, naive B cell undergo a clonal selections and expansions and differentiations into plasma cell and memory cells. So because, for example, there's antigen A invade our body system, so this is the first time antigen A being introduced to our body system, and it will be recognized by naive B cells. Okay, so it needs time to the naive B cell to undergo clonal selections and differentiations to plasma cell and memory cell. So that's why they have a lag period here. Okay, the lag period is followed by a logarithmic increase in serum antibody level, which reach a peak periods for a variable time and the decline. So it means that after plasma cell secrete antibody, the concentrations of antibody in the serum will be increased, right? And this shows that there is a response and produce the anti-A, all right, anti-A to the antigen A lah. Okay, and it will took like nearly 14 days, all right, longer time. So after everything is being neutralized, the concentrations of the antibody in the serum will be decreased. Okay, so the first attack, there is a lag phase to give time for naive B cell to undergo proliferation and differentiations into plasma cell, and then plasma cell start to secrete the antibody in the serum start to be concentrated, and the antigen is being neutralized, then the concentration of antibody is reduced. The durations of the lag times varies with the nature of the antigen, of course. So if the antigen is very dangerous or have a higher toxicity, it will take longer time on the lag period. The antibody isotope is primary immune response is mainly IgM. Okay, during the first infections, right here, right, mainly the plasma cell will be secret IgM. So kat sini IgM lah yang akan bekerja. Alright, IgM. Because IgM is a pentameric in structure. There are 5 antibody that clumping together as a group. Okay, during this time, memory B cell form during primary immune response stop dividing and enter the G0 phase of the cell cycle. This cell have a variable lifespan. So lepas dah everything settle down, right, memory B cell will be kept in latent mode. Senyap lah. It's not dividing at all. But then, what happened if the same pathogen invades into our body? Okay, for the second time, is the response will be in different way, right? Why? Right? Compared to primary immune response, the secondary immune response, the way that secondary immune response react is totally different, right? Why it happened? This happened because during the first infection, not only plasma cell being produced, okay, memory cell also is being produced to memorize the nature of the antigen. Okay, and the response result from activations of the memory cell. So when there is, okay, just ignore this antigen B first. When there is a subsequent infections, okay, memory cell will be triggered. Okay, they differ both qualitatively and quantitatively, of course lah, because untuk second infection ni tadi memory B cell dah sedia ada, right? The memory B cell is there. Okay, so bila antigen masuk je, memory B cell dah recognize, right? So bila memory B cell recognize the foreign antigens, it will stimulate the plasma B cells. Okay, the amounts of antibody produced after the first encounter with the antigen are smaller than the amounts of antibody in secondary immune response. So in the secondary immune response, bila memory cell dah recognize the antibody is there, alright? So bila the antibody is there, the plasma starts to secrete antibody in the large amounts of concentrations. Okay, so banyak antibody is being produced in the second infections. So if you can see here, there is a larger gap between the primary immune response and the secondary immune response. Okay, so in secondary immune response is much higher. Sebab apa? Sebab dia dah ada memory from the first infections. And the elevated concentrations of antibody, if you can see here, is much shorter compared to the primary infections. Okay. Right. Secondary response also show increase in antibody infinity. Infinity means that affinity means that the antibody yang being produced by the plasma cells is very specific. So dia dah kenal dah the first antigen tu tadi. So that's why the binding of the antibody with the antigen A senang saja. Okay. The lag period is shorter than in the primary response, of course, and the antibody isotope in the secondary immune response is IgG, and under certain situations is IgE and IgA. So just now in the primary immune response, mainly IgM is being produced. Okay, but then during the subsequent infections, IgG will be produced. Okay, so kalau you tengok dekat sini, okay. In the diagram, not only antigen A, antigen B also is being introduced. Okay, so antigen A untuk subsequent infection, there is already a memory cell. So memory cell dah recognize antigen A. But then how about antigen B? There is no memory cell for antigen B. So that's why the graph for antigen B is similar or to the primary response. So sebab dia tak ada memory cell lagi kan. So the pattern of the graph is the same as the primary immune response. Okay, but then if there is a subsequent infections on the antigen B, baru the graph dia sama macam antigen A ni tadi. Okay, so this is the summary. You boleh tengok ni nanti, the summary of the primary antibody response and secondary antibody response in infographics. So it's much easier to understand. Okay, the comparison. Okay, kalau you tengok ni comparison, right? The lag period following antigen administrations. Primary response generally 4 to 7 days, right? Longer time. But then in secondary is very fast, between 1 to 3 days. The antibody is already been produced. Okay, so sangat cepat. Right. So we already covered the humoral immune response. Now let's go to cell-mediated immune response. Okay, remember, on humoral mediated immune response, it's only involve B cells, right? With a help from T helper cell, of course. But then in cell-mediated immune response, it's always only involve T cells, right? Both T helper cell and T cytotoxic cell, right? CTL. Okay, let's go to cell-mediated immune response. Okay, so this is the phases of cell-mediated immunity. It's almost the same as a humoral mediated immunity. First, recognitions of cell-associated microbe by T cells. Second, activations of T cell lymphocytes. The third one, clonal expansion and differentiation. And the last one is effector functions. Okay, let's go to the first function. Let's go to the first factor, which is recognitions of cell-associated microbes by the T cells. Okay, both cell TH is T helper cell and TC is cytotoxin T cell. Okay, can recognize antigen only when it has been processed and presented on the cell membrane of an antigen presenting cell, APC, or target cells, okay, respectively in associations with the cell MHC. Okay, if you can remember from the last lecture, right, previous lecture, all nucleated cells, all nucleated cells have MHC1, okay, but then APC have MHC2, okay. So MHC measure measure histocompatibility, okay, as a receptor that can present the antigens on the surface of the cell. Okay, antigen presenting cell is the cell which present antigen to CD4 T helper cell, whereas the target cells, the cell which present antigen to CD8 TC cells. Okay, kita talk di sini. Right. So this is the target cell. Right? Target cell can be all nucleated cell lah. Selnya ada nukleus sahaja, nucleated cell, ada MHC1. Whereas on antigen presenting cell, APC have MHC2. Okay? On MHC1, it only can be recognized by cytotoxic cell. And the consequences of the recognitions, the target cell will undergo lysis or apoptosis. Okay. Whereas in APC, it will recognize the MHC2 will be recognized by T helper cell. Okay, and when the consequences of the engagements between MHC2 from APC and CD4 from the helper T cell, it will activate a macrophage. It will activate a clonal expansions of T cells, and also will activate humoral immune response. Okay, ada tiga consequences bila T helper cell recognize antigen on MHC2. Okay, right. So, what is ini? Okay. MHC is also known as a human leukocytes antigens. This process is called antigen processing and presenting. Okay, what is processing and presenting? It's like processing is a protein antigen is degraded into a peptides. Okay, for example, a virus or viral DNA is being invaded into host cell. Okay, then the viral DNA will be degraded and processed into a small fragment so that dia boleh present dekat dia punya surface. Same goes to the APC. It will degrade the antigens from the outside, alright, exotoxin masuk phagocyte. Then it will process the antigen, degrade the antigens into small peptide, and the small peptide will be presented into the surface. So itu nama dia proses. And presentation associations of peptide with MHC and transportations of MHC peptide complex to a cell membrane. So lepas dia dah degrade, baru dia present dekat luar. Lepas dia dah degrade antigen ni, baru dia present dekat luar. Okay. Okay, cells which have been induced to function as APCs are as follows. There is a macrophage, there is a B lymphocyte, and there is a dendritic cell. So all of these three are known as a APC. Okay, remember class, cytotoxic cell, T cytotoxic cell, also known as CD8. Okay, T helper cell, also known as CD4. What is CD8, CD4? CD8 and CD4 is a co-receptor. Okay, it's very specific. CD8 only can recognize MHC1, whereas CD4 only can recognize MHC2. So that's why T cytotoxic dengan T helper cell dia tak apa to say, the activation is not redundant lah. So the means that APC cannot be recognized by cytotoxic T cell, and the target cell cannot be recognized by T helper cell. Okay, activations of T lymphocytes. Antigen recognized by TCR signals. This is TCR. Okay, TCR trigger signals that are delivered to the interior of the cell. The signal are delivered by CD3. This is CD3, right? CD3. This is TCR. This is CD3. Sama juga dekat T helper cell ni. And zeta protein as well as the co-receptor either CD4 or CD8. Co-receptor recognize class 2 or class 1 MHC molecule. CD4 recognize MHC2 and CD8 recognize MHC1. Okay, in response to antigen, T lymphocyte, especially CD4 T helper cell, rapidly secrete different cytokines that have diverse activities. Okay? The first cytokine to produce within 1 to 2 hours after activations is interleukin-2, IL-2. Main action is to stimulate the proliferations of the T cell. Okay. So, ni gambar summary lah, but then I just want to show you. So, bila T helper cells is being activated, means that they dah recognize macrophage, for example. So bila dia dah recognize macrophage, it will secrete out the cytokines. I mean the cell signal. Actually banyak. There is many cytokines. Ada interleukins 1, interleukin 2, interleukin 12, 14, so on and so forth. But then in here, to be specific, it will secrete interleukin 2. So when it secretes interleukin 2, it will help cytotoxin T cell to differentiate. And helper T cell also will be differentiates into memory helper T cells. It auto differentiates. Okay, CD4 T helper cells may differentiate into diverse effector cells that produce distinct sets of cytokines that perform different functions such as. Okay, just now CD4 T helper cell will secrete out interleukins to active interleukin 2 to activate proliferations of T cells. Okay, so ini the other cytokines, another cytokines that been secreted by T helper cell. The first one is it will produce interferon gamma, which activate phagocytes and stimulate the productions of opsonizing and complement binding antibodies. Okay, so in here, kalau you boleh tengok kat sini, it activates cell-mediated by phagocytosis and also at the same time stimulates humoral immune response by opsonizing and complement binding antibody. Okay, so that's why I told you at the beginning, CD4 T helper cell is very important because it can mediate, it can activate both humoral immune response and cell-mediated response. And T helper cell also will produce interleukin-2, which stimulate the production of IgE, okay, and produce interleukin-5, which stimulate eosinophils, which function mainly in defense against helminths. So this is untuk parasite lah, okay. Whereas CD8 cytotoxic T cell, or known as CTL, recognize peptides or intracellular protein antigens and may require help from CD4 T helper cell to differentiate into effector CTL. Okay, maksud dia macam ni. Yeah, okay. So, So, T cytotoxins only recognize intracellular protein antigen. Intracellular protein antigen means that from macrophage ni tadi, so from infected host cell ni tadi, okay, so it means from intra, dalam lah. Okay, yang ni from exo, luar, dia masuk dalam. Tapi this one memang dah daripada dalam, dia dah degrade viral DNA tadi, dia dah presented, baru cytotoxin tu recognize. But then, when it recognize, dia tak boleh proliferate lagi. It needs help from the T helper cell to differentiate into effector CTL, cytotoxin T lymphocytes. Okay? Right. Next is effector functions. Right, the last one is effector functions. There's two types of cell-mediated immune response. The first one is CD4 T helper cell activate macrophage. Okay, so T helper cell tu tadi bila dah recognize, it will secrete out, for example here, interferons gamma, and will activate phagocytes. That can be done by macrophage to kill ingested microbe and produce substance including reaction oxygen intermediates nitrate to kill microbes and produce cytokines that induce inflammation. So bengkak lah. And the second one is effector CTLs. Okay, effector CTL. CTLs tadi, already undergo proliferations and differentiation lah. Kalau tidak, known as CD8, right? Cytotoxic T cell. But now, after it been undergo proliferation and differentiations, it known as effector cytotoxin T cell lymphocytes. Kills cell harboring microbes in their cytoplasm. CTL induce cell death via two mechanisms. So there is two mechanisms that can be triggered by CTL. Right? The first one is prophorins or granzyme pathway, and the second one is FASL pathway. Okay, what is granzyme pathway? Granzyme pathway, the primary event in CTL-mediated death in the granzyme or prophorin pathway are dia punya mechanism lah, but then I will explain it in detail in this diagram. So the first one, when CTL engage with the target cell. So the target cell been infected, for example, by virus. The viral DNA already been digested, all right, and the small digested DNA will be presented into the surface, and CTL recognize it. Okay, so when the CTL recognize, both of them will be conjugates. Conjugate means that will be, respond to each other lah. Okay, and it will start to produce a granule. So granule ni tadi ni lah, granzyme. Okay, it's contain granzyme, and the CTL cytoplasmic rearrange, right? Rearrange. And the CTL granule exotosis, right? Exocytosis means that the granule will be exocytosis into the target cells. Okay, bila dia dah masuk dalam target cell, then the target cell will be undergo apoptosis. Okay, and with dissociation, lepas dia dah trigger the target cell to undergo apoptosis, it will dissociate and the CTL will be recycled back. Alright, the whole mechanism will be repeated again to the another target cells. Okay, the second one is FASL pathway. Okay, FASL ni is a type of ligand. FASL is the transmembrane protein that can deliver a death signal when cross-linked by its natural ligand. Okay, FASL ligand which are found on the membrane CTL. So this is CTL. Right, this is CTL. This is FASL ligand. And this is the target cell that have FASL receptor. So bila dia bertemu, alright, the link engage together, and the target cell will undergo apoptosis. Okay, let's go to the different differentiations between humorals and cell-mediated immune response. So ni yang gambar summary I show you just now. Okay, let's go to this one. Okay, this one. Okay, so this is the differentiate between humoral-mediated immunity and cell-mediated immunity. And in humoral-mediated immunity, the mechanism will only involve antibody, whereas in cell-mediated, this only involve cells. Okay, and the cell type is B lymphocytes. For the humoral and cell-mediated, it will involve T lymphocytes. The mode of action is antibody is circulating in the serums, whereas in cell-mediated, with direct contact between target cell and the T cell, okay, and the production of cytokines. Okay, the purpose of humoral is primary defense against extracellular pathogen, extracellular bacteria and circulating virus, whereas on the cell-mediated immunity, the primary defense against intracellular pathogen, virus and the fungal intracellular bacteria, also tumor antigens and graft rejection. Graft rejection ni lah, betul tu transplant and tumor antigen macam cancer cell. Cancer cell ni kan dia easy to proliferate. Okay, so the cancer cell, the cell that dah jadi cancer, it will represent the weird protein that been expressed in the cell on the surface, so that T helper cell akan recognize, T cell akan recognize. So bila T cell cytotoxin recognize, dia baru induce cancer cell tu tadi to undergo apoptosis. Okay, so yeah, you can test yourself by these questions. There's 3 questions. If you can answer all these questions, it means you already know completely about chapter 5, the immune response, the differentiation between humoral, cell-mediated, and the antibody response, as well as the effector function of antibody. Yang ada 5 groups tadi. Okay. So I guess I stop here. If you have any questions, freely to ask me through WhatsApp. So you can drop your comments or questions on the Google Class, okay, or onto my YouTube account. Okay. So yeah, I hope to meet you again on the next class. So Assalamualaikum warahmatullahi wabarakatuh. Terima kasih.